This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 198, recorded on May 30th, 2019. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
How is everything?
Good. I'm teaching a new class of first-year medical students about the wonders of bacteriology.
Great.
Hey, you're the
8 a.m. 8 a.m.
You're
every
the person.
day.
Oh, my. You're going to have to take attendance. Otherwise, they're not going to be there.
Also joining us from Small Things Considered, Elio Schaechter.
Hello there.
Hello, Elio. How's the West Coast? Is it still there?
Yeah, still here. The weather is completely crazy. We've had a lot of rain,
It's
which
good.
is unheard of.
But that's good, right?
Oh, yeah, it's good for them, but it's strange, and it's kind of cool. Strange weather.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
It's not cool there, is it?
No, unfortunately, right now it's a whopping 33 degrees centigrade or 91 degrees Fahrenheit. It's
Ouch.
quite toasty. And the humidity will let you know that you're not in a dry oven. You're in a moist oven.
We had here in New Jersey a tornado watch the other night.
Oh, my God.
They told us to go in the basement
Oh,
for about an hour. And so we did.
I was at D.C., Reagan Airport in D.C., and the tornado bells went off.
It was a couple
and there
days
was
ago.
a tornado warning.
Was that
So
recently or what? Or was
It was a week ago Thursday. Yeah, a week ago
Oh.
today.
Hmm.
So I found like a stairwell and just hung out there.
Wow. It's weird. We usually don't
Crazy.
have tornadoes here. It's very strange. Before we get into our science today, if you like what you hear on TWiM, consider supporting us financially. You can go to microbe.tv/contribute, and you could, for example, give us $1 a month. And that way you can feel great about supporting real information on the internet, science. All right.
You didn't say what number TWiM this is.
198.
198. Wow.
Our next one, which will be 199, will be at ASM Microbe.
Yeah.
ooh.
Unfortunately, I won't be there.
I tried to make it to be 200, but it didn't work out. So we will have a 200 afterwards.
Let's all go to San Diego and do our 200th from
Hey,
Elio's place.
I like that.
Hey, yeah, we could go down after Microbe, right?
Yeah.
That would be fun.
Sure.
Be a lot of fun. How far is
Take this,
San Diego's like days from San Francisco, right?
It's an hour and a half, something like that.
By plane?
By plane.
Mm.
Not much
But
like-
didn't the governor build his special bullet train? Can't we take the bullet train?
Let's not get started on that one. That's a hot
Oh.
topic.
That's a sore subject.
Sounds like it. All right, let's talk about some science. Elio has a snippet for us.
All righty. It's a snippet that kind of an odd subject. I think many readers know what rickettsia are. They're the agents that cause typhus and related diseases. And they are considered to be strict intracellular parasites in the sense that they multiply only within host cells. Okay? That's it. Sort of been a tenet of microbiology that seems unshakable. Well, it got shook because people have found rickettsia, albeit of a different type than the usual ones, which grow outside of cells. The paper is called Deianerea, that's a new term, an extracellular bacterium associated with the ciliate Paramecium, suggests an alternative scenario for the evolution of Rickettsiales. And it's by about 11 authors, so I won't mention any but the first one, which is Michele Castelli. The second one is Elena Sabayeneva. The last two are Davide Sassera and Giulio Petroni. And they are a collection of people from St. Petersburg, Russia, and from three Italian universities, Pisa, Parma, I believe, and Milan. And what they found is an unusual story. Let me tell you how it started. They got a culture of Paramecium, which they can keep in very nicely in culture. You feed Paramecium bacteria, that's what they eat on. And this Paramecium, after a month in the lab, showed many dead cells with abnormal phenotypes, or different shape, and non-motile because they completely lost their cilia. But what they saw in the microscope is they were surrounded by a dense layer of tightly packed bacteria, about 10 bacteria, 10 deep, give or take. Very unusual. It turns out that there are other ciliates that have arrangements like that. They're surrounded by bacteria, chlorella name. But it's unusual. So these are bacteria which are growing outside of cells and obviously multiplying and causing the death of the Paramecium. Let me describe them first. They have a double membrane cell structure like Gram negatives and are curved rods up to 1.5 or so microns long and very thin. And they appear narrow on one end with a sort of what they call an apical tip, which is in direct contact with the outside of the Paramecium cells. And a few of them show that they're dividing. They gave these guys, so they did what is done these days. They did a thorough genomic analysis, and they call them Deianerea vastatrix. The term has to do with a mythical Greek mythology. Deianira was the wife of Hercules, and she, by mistake, killed him by giving a poisonous shirt. So this is a poisonous shirt around the Paramecium, all right. Anyhow, Deianira is a little bit unusual. So how does the Paramecium survive? Well, if they die from being surrounded by this bacteria, how come they're around at all? Well, it turns out that if you sort of transfer them into bacteria, a medium that have the food bacteria, they will survive. In other words, it's like they shake loose their coat of the parasite. That's how they survive, and you can study reinfecting them. So anyhow, so what do you know about this bacteria? 16S RNA has highest similarity with a bunch of uncultured Rickettsia-like bacteria. So there are bacteria which are Rickettsia-like, and there's one that's very close. It comes from an earthworm. And they made a specific probe, and they found that these bacteria are not present inside the ciliates, ever. Not in food vacuoles, not in anything. And they are a divergent family-level clade, together with 14 other uncharacterized database sequences. So these are uncharacterized bacteria, mainly from aquatic environments, in some cases associated with eukaryotes. And this family has a sister relationship to the Anaplasmataceae, which is one of the clades of the Rickettsiales. But it's different. So there are 3 clades in the Rickettsiales. This is a 4th one. The genome of this bug, Deianerea, I learned to say it yet, has a single DNA, circular DNA, 1.2 megabases long, and with a 33% GC contact, which is typical of the known range of the Rickettsia. What does it code for? So here's a bug that is supposed to be a cousin of a strict intracellular parasite, and in fact, it's not. It's not a strict intracellular parasite at all. So you'd expect it to be more competent at free life, because Rickettsia, the typical Rickettsia, when you look at the genome, it's clear why they don't grow outside of cells. They lack an awful lot of biosynthetic capacity. So How about these guys? What do they code for? They have complete like gluconeogenesis and a pentose phosphate pathway. They have a Krebs cycle and OXPHOS. They can carry out oxidative phosphorylation. They lack cytochrome C and its oxidase. And they have the biosynthetic pathways you'd expect for the main structural components of the cell inner membrane, cell wall, and the outer membrane. Now, they don't have all the nucleotide synthesis capacity, so they need and only make certain cofactors. They make biotin, heme, ubiquinone, folate, and the iron sulfur cluster. So they lack some things, but they get them because, like all Rickettsiales, they have multiple membrane transporters that probably complement the metabolic deficiency. In other words, they are fed by the paramecium for a number of these things. Interesting is that they make 16 out of 20 amino acids themselves. This is strikingly different with the rest of the Rickettsia, which have many fewer than that. In fact, the total genes devoted to amino acid production in these guys are 56, whereas no other member of the Rickettsia comes close to 30 genes. Okay, so there you have it. They are not totally capable of free life, and in fact, it's even though the authors don't deal with this, it's not likely that you can cultivate them outside of the paramecium, because they need something from the paramecium. Not as much as the Rickettsia of typhus, which need a lot more and therefore are strict intracellular parasites. So the Rickettsia are interesting for an awful lot of reasons. One of them, not only they cause interesting diseases and important diseases, but also they are supposed to be a sister group of the mitochondria. So the idea was mitochondria arose by a cell, primitive primordial cell, swallowing a Rickettsia, and the Rickettsia becoming degraded genomically into the least that it needed to do, namely to become a mitochondrion. Now, this point is very nice, and however, it's highly debated.
I was going to ask you, Elio, is this microbe aspiring to be a virus or a mitochondrion?
It's, I would say, certainly not aspiring to be a virus because it has an awful lot of metabolism going for itself. No, I think it's probably aspiring to become a free living cell.
Oh, it's going
free living in the sense that we can grow without the paramecium. That's my guess.
It's difficult to say, though, isn't it? We just have a snapshot and we don't know which way it's headed.
Exactly. Yeah. And there's a lot of discussion. If for those of you who care about the subject, there's plenty in this paper. It is rich in the consideration of the possible evolution of these guys, as well as of all the Rickettsia. So they pose an alternative scenario. They feel that these guys, they started as extracellular cells, like they are now. But the Rickettsia, and so did the rest of the Rickettsia, but the rest of the Rickettsia became intracellular, strict intracellular parasites. Notice, however, that although they're strict intracellular parasites, they have an extracellular phase. They go from cell to cell via the bloodstream and things like that. So they're not always, Rickettsia are not only intracellular. These guys are apparently never intracellular. There's a lot of discussion possible, but notice the interesting thing. Rickettsia enter the cells, the regular Rickettsia, by inducing phagocytosis. And they escape a phagosomal membrane to become living freely in the host cytoplasm. And they're also interesting, they exploit the host cytoskeleton for movement. They take actin at one end of the cell, and the actin gets polymerized more and more, pushing the Rickettsia around. So the Rickettsia run around the whole cytoplasm of the cell. So they feel that given all this and putting it all together, they feel that the proto-rickettsia may have been extracellular, and they probably had adaptation mechanisms to living with eukaryotic cells, such as having a type 4 secretion system and the ability to translocate ATP and ADP. Rickettsia typically do not make ATP and ADP, so they need it from the environment. That's kind of unusual. Not many bacteria do that. So the idea is that the original rickettsia were extracellular. They became more and more intracellular prone by having a tighter and tighter association with the host cell and a reduction in the genome. So that's the message. It's really, let me say that the paper is worth looking at because the electron micrograph of these Paramecium covered with bacteria are really quite unusual.
I was gonna raise that. I looked at that figure 2, that first panel, and it reminded me of Linus from the old Charlie Brown series, where he'd flick his blanket and all these fleas would come flying away.
It does.
It looks like this Paramecium is just outnumbered by 100, 1,000 to 1. It's amazing.
It's something like 1,000 to 1. As I say, the layer of Paramecium are quite big for unicellular organisms, and they have this layer which is about 10 bacteria thick, maybe more.
Wow.
It's really amazing. That figure is just like nothing I've ever seen.
I wonder how the Paramecium benefit.
Does the Paramecium benefit? The Paramecium doesn't seem to benefit. If you leave it alone, it'll die. If you don't wash the bacteria away, it'll die. So I'm not sure this is to the advantage of Paramecium. Hard to imagine what's in it for it.
Well, just before I came to TWiM this afternoon, I was over at the infectious disease conference that's held every other week. And today they were talking about C. diff. And, the C. diff microorganism attaches to epithelial cells. And I had just finished looking at this paper, and I'm going to myself, well, our Paramecium, the precursors of epithelial cells, and what I'm seeing is the equivalent of what C. diff is doing to people when it causes invasive disease through its toxins. And, it's just, the epithelial layer breaks down and you get leakiness, which is the whole issue with C. diff pathology. But looking at this, it begs the question, what is the rickettsia doing for this particular Paramecium? And, how do you begin to...
The answer is not much. The answer seems to be not much. It seems to be a real parasite. But like C. diff is.
Elio, if you isolate Paramecium from the wild, are they covered with these bacteria? Or
no, this is a characteristic of a Paramecium called Primaurelia. It's not Paramecium aurelia. It's a different Paramecium. So but I don't know the answer. Does Paramecium aurelia also get covered? We would have known. I mean, with the amount of work that's been done with Paramecium, the fact that this is news means it's unusual. Otherwise, we would have seen it.
I'm not clear whether this is something created in the laboratory or you can...
Oh, no.
So how do we know it exists out there?
Oh, oops. No, I shouldn't have said that.
So you can isolate these Paramecium and you find rickettsia on them, right?
Yeah.
That must be. Okay.
That's how they found it.
And you said
they got a strain of Paramecium Primaurelia, and after they mounted the lab, it started to die.
I see. And they're covered.
That's all we know.
All right, and they're covered with the diarrhea. And if you take them off,
good for you. You said it right.
If you take them off, then the Paramecium survive. Is that correct?
Right. That's right.
Okay. If you shake them off like
Pigpen's blanket.
Linus, thank you.
No, it's Pigpen.
Oh, it's Pigpen. That's right. Pigpen.
Pigpen.
Now, Elio, in these electron micrographs, they're attached to the Paramecium. Some of them are by stalks.
The innermost layer.
Yeah. So are they thought to be grabbing components from the Paramecium that way?
That's right. That's right. Since they do not have a complete metabolic spectrum, being able to make everything themselves, they need a few things, and they get them from the Paramecium, obviously. no other place.
Yeah, and probably as they acquire the capacity to scavenge, then they those biosynthetic pathways deteriorate.
Yeah.
That's right.
If you don't use it, you lose it.
Very good.
Yeah.
But not every bacterial cell is got a stalk, right?
That's true.
Just
That's true.
So I wonder if it's a dynamic process. They're forming and breaking and so forth, and so they can share the resources. Would be interesting to see some live microscopy to see if you could see that.
That's true. I'm surprised they didn't do that.
Yeah.
Yeah. There's not much.
More work.
Weird and wonderful biology out there.
Yeah, very surprising. I gotta tell you, I cut my teeth with rickettsia way back. When I got drafted into the Army, this is after I got my PhD, I was sent to a lab at Walter Reed Army Institute of Research, and my boss was a famous rickettsiologist by the name of Joe Smadell. And he was bothered by the fact that at that time, people didn't really know what to call rickettsia. Should they call them bacteria? Should they call them viruses? So he says, find out once and for all. So they gave me some tissue culture cells infected with rickettsia. I put them on a slide on a warm stage under the microscope, and I looked until I had to recognize which of the rickettsia, which turned out to be easier than I thought because they're quite a bit bigger than mitochondria, actually, and darker. So I could tell which are the rickettsia, and I would look and look and look and take pictures every minute until one divided into two. And when that happened, that solved the question. Are they bacteria? Of course they're bacteria. They're not like viruses because viruses fly apart and then reconstitute. But these guys never fly apart. They are cells.
Elio, let me share that I was preparing for my rickettsia lecture this morning.
Oh, really?
Which is the first time I gave this one. I was looking through some reviews and I came across a reference to one of your papers. Schaechter et al.
Is that right?
Yep.
That makes my day. Thank you.
That's cool. Well, I notice in this Rickettsia, there's I don't know, is this a family? The ACA, I guess those are families, right? Rickettsia is a family level.
Yeah.
There's a Midichloriaceae.
Oh, yes.
The Metachlorians from Star Wars.
Must be where George Lucas got the name, right?
No,
they named it. They named it from Lucas.
You think?
Yeah.
Well, we got to go back and look at the taxonomy to see when they named it. If it's post, 1983, then it's probably from Star Wars.
Okay.
It is from Star Wars. I know that.
You do? Okay. That's interesting. All right. Yeah. That's cool. Thank you, Elio.
My pleasure.
All right. For a paper, we have an article from Science. It's called Bacteriophage Trigger Antiviral Immunity and Prevent Clearance of Bacterial Infection. And this is from a large group of authors from Stanford University, Baylor College of Medicine, University of Washington, University of Montana, and the Palo Alto Veterans Institute of Research. And the first author is Johanna Sweer, and the last corresponding author is Paul Bulicki.
I counted 23 authors.
And this is pretty interesting.
It's downright fascinating what they have done here, and it's going to literally set phage therapy on its ear. Just a spoiler alert.
Hold that thought.
Yeah.
Yes, that's a good one. We'll come back to that. So this is about Pseudomonas aeruginosa. Gram-negative bacterium causes a lot of morbidity and mortality. You can find it in wound infections, ulcers, and burns. It's often antibiotic resistant. There's no vaccine. So this paper looks at the role of a phage, a filamentous phage, which we will call PF bacteriophage, which are made in a temperate fashion by infected bacteria. These phages don't lyse the bacteria, but they integrate as a prophage, which is passed on as the cells divide. And when filamentous phage are made, they're extruded from the bacteria, but the bacteria don't lyse. And what they're asking in this paper is, does this phage play any role in the pathogenesis of Pseudomonas infections. And the first thing they did was get 111 patients with infected, non-healing wounds who came to the Stanford Advanced Wound Care Center. And they identified 37 of them who had Pseudomonas aeruginosa infections. And then they asked, how many of them have PF phage in the wounds? So they found using PCR to measure. Now they're measuring the prophage, which is integrated into the bacterial genome. 68% of these PA-infected wounds had prophages, 25 out of 37 patients. Okay, so
the old wounds. And
the older wounds had a higher percentage of phage than younger wounds. Okay, and so there's some association of this phage with chronic wounds in this particular cohort. Okay, so now we know there's a phage in a good fraction of these wounds. It's associated with chronic wounds and longer lasting wounds, which, by the way, means older than 3 months.
Mm-hmm.
So can the phage actually promote infection? So they have a mouse model that they use to study this. They make a cut, they wound the mice, they then inoculate it with luminescent PA, so they have a gene in there that's going to luminesce so they can easily see it, and they measure bacterial infection by quantifying luminescence.
And this isn't a special Pseudomonas. This is the garden variety lab strain PA01.
Right.
So there's nothing mythical or magical about their particular Pseudomonas strains other than the fact that they put a GFP and marker in it.
Right. So they have a strain which has bacterial strain PA01. As Michael said, this has the prophage in it. And in their mice, they can measure the copies of, there's 10 to the 9th copies of the phage per wound. And if you compare that bacterium with one lacking the prophage, there's no difference in growth rate. There's no difference in luminescence comparing the two. But let's compare pathogenesis, infection rate. How many bacteria does it take to establish a wound infection? And here we see a big difference. If you look at the number of bacteria where 50% of the wounds become colonized, you need a lot more bacteria if they're lacking the phage.
Like a couple logs more.
Yes.
100 times more.
This is what terrified me from the perspective of wounds, that a simple prophage, I mean, those of us old enough remember the famous filamentous phage M13. And this is probably no different than an M13 phage infecting E. coli. It just happens to be a Pseudomonas phage. And this is remarkable that it's able to drop the infectivity rate logs, not percentage, logs.
100-fold.
100-fold, yeah. So then if you put the phage back into the strain, the bacterial strain that lacks it, it makes it now have the more efficient infection rate. So in other words, the phage reduces the number of bacteria that you need to establish an infection. What about disease and death? When they use a higher dose, so far they've been using a low dose of bacteria. It doesn't kill the mice. But when they up the dose, you get all the mice infected. And the mortality is higher in the mice that have the bacteria containing the phage compared to without. If you look at the graph, without the phage, 100% of the mice survive. With the phage, it looks like 75% are surviving. It's not huge, but it's different. And
it's just significant too.
Yes.
304.
And the mice infected with bacteria containing the phage, they lose weight. They lose more weight. So this having the phage in there promotes the incidence of disease and the virulence of the wound infections in the mouse model. All right. So what's going on? Now we have the baseline. What's going on? What's controlling this? So they say, is there some difference in the immune response? And they look at immune cells that are infiltrated into the wound, and they don't really see any difference between the two bacterial strains. But they look at uptake of bacteria by phagocytosis. And they measure uptake, and they found that having the phage in the bacteria causes a reduction in the uptake of the bacteria by dendritic cells and macrophages. These are the typical phagocytic cells that would be clearing infection. And they also showed that if you take a clinical isolate of Pseudomonas aeruginosa, having the phage also reduces the uptake by these phagocytic cells. And in fact, if you take this bacteria lacking the phage, put the phage back in, that is enough to inhibit phagocytic uptake. And in fact, adding the phage to phagocytes will inhibit the uptake of E. coli, a different bacteria altogether. So somehow
that's where it gets really interesting.
The phage is messing with the phagocytes, and it's preventing them from taking up the bacteria.
I'd just like to interject here. I spent 2 years of my life trying to engineer phage to persist in the bloodstream so that they would be able to seek out an infection and attack it. And little did I know that the phage would actually, if this model holds true for other things, I mean, it's scary.
That's right. You could be doing the opposite of what you hoped. Yeah.
Yeah.
All right. So why is phagocytosis inhibited by the phage? Well, we know that a cytokine called tumor necrosis factor is important for uptake of these bacteria by phagocytes. So they ask, does the phage affect the production of TNF? And you can do that by taking phagocytes from mice. You add LPS or an algal derivative. These will induce TNF. If you add the phage or a control, PBS control, what you find is that the phage inhibits the production of tumor necrosis factor in these phagocytes. Just adding the phage to the cells, pre-treating the phagocytes. And if you add back TNF, it will now restore the ability of the phagocytes to take up the bacteria. So it's clear that the phage is somehow affecting the production of tumor necrosis factor, which in turn is needed for phagocytosis of the bacteria.
It increases the rate of phagocytosis, makes them much more efficient.
Right. So what's going on? Well, basically, they find that these cells, when you stimulate the phagocytes to produce TNF, you get production of mRNA, but you don't get protein synthesis. You don't have the production of TNF protein. So somehow the phage is inhibiting translation of the mRNA for TNF. And they say, well, one way that this could happen is through interferon. So let's look at the interferon pathway. And so they take a number of mice that lack different components of the interferon response. And the main one here is mice that lack the type 1 interferon receptor. And in phagocytes from those animals, the phage does not reduce TNF production, and it does not impair phagocytosis. So that tells you that the phage is impairing TNF production in a manner that depends on type 1 interferons. And then they use a series of other genetically altered mice to show that it is actually the toll-like receptor 3 that is responsible for sensing the phage, and that leads to inhibition of TNF production. So toll-like receptor 3 is one of a number of membrane proteins that sense microbial patterns. They can be nucleic acids, they can be other components, and they initiate innate immune signaling. So somehow this phage is activating toll-like receptor 3 and causing the inhibition of TNF translation.
That's a long way of saying it's bad news.
Now, interestingly, the TLR3 normally detects double-stranded RNA, but this phage is a single-stranded DNA phage. So they're scratching their heads. They're like, well, how does this phage activate TLR3? So they found that if you transcribe the phage genome and get RNA and add that to cells, that will activate TLR3, which is what you would expect because it's sensing RNA. If you add the phage to cells, that activates TLR3. And the phage appear to be taken up by the cells, and they believe that the phage DNA is actually transcribed in the eukaryotic cell, in the macrophage, and the RNA produced is sensed by TLR3, and that results in inhibition of...
But it's not double-stranded, is it?
No, but remember, messenger RNA likes to pair with itself. And the most telling statement in their paper, however, our data suggests that a natural bacteriophage can produce mRNA within human cells. Because there's no bacterial ribosomes in a eukaryotic cell, that mRNA is just able to pair with itself, and it can effectively serve as a facsimile for double-stranded RNA.
That's their deduction.
That's what they think. Yeah.
Yeah.
I mean, there are probably cryptic promoters in the phage genome which are transcribed, in the eukaryotic cell, and then the mRNA could either base pair with itself, it could form double-stranded stretches, or in some way become double-stranded. So that's plausible, I think. And
they do show some lovely transmission EMs where the phage are co-localized within endosomes with TLR3. And also they see it out in the cytoplasm. So
yeah, it's quite interesting.
It's in the right place.
When you add the virus to the phagocytes, they get taken up in what's apparently a receptor-independent fashion. They don't actually bind to anything. They're just taken up, and they can see
it's a particle.
The particle, yeah. They're particles. They're taken up, and as Michele said, they can co-localize them with markers in endosomes. With TLR3 is actually in endosomes. It's in the lumen of endosomes, and they see that the phage co-localizes with it. So that's pretty convincing that it's being taken up, and somehow it's being transcribed. The DNA must be getting out into the cytosol and being transcribed. And this can happen in a diverse range of cell types. They look at a variety of splenocytes and lymphocytes and different cells that would infiltrate the wound, and B cells, dendritic cells, mononuclear
NK
cells. A whole bunch of them happens in all of them. This idea that the virus gets in and is transcribed, and it inhibits the production of TNF, which impairs phagocytosis. And so their model is the virus gets into cells, it triggers TLR3, the interferon is made, it inhibits TNF, and that limits the phagocytosis of the bacteria, and therefore you get long-term infections.
So is it an oversimplification to think that the leukocyte is confused? It's getting mixed messages. It's just sensed that there is something in the cytosol, in the TLR3 compartment, so it wants to switch to this cell-mediated immune pathway interferon, and therefore it dampens the pro-inflammatory TNF-alpha pathway.
Yeah,
I think
so.
it's evolution writ large, Michele. I think it's just showing the elegance of the human immune system, or the immune system in general, being able to react to these very clever pathogens, whether they be virus or bacterial.
Of course, it's RNA that's sensed, right? So
yes, it's the RNA that's sensed.
So the innate system is designed to detect RNA, among other things, and respond. And I think perhaps the phage bacteria combination has evolved in a way to, I don't want to say take advantage, but to somehow benefit by that, right?
Co-opt, I think, is the word.
Co-opt, maybe, or it's too active, but you know what I'm talking about.
Yeah.
So it finds itself in a situation where TNF is inhibited, and that's beneficial. The bacteria can stick around longer. The last experiment they do involves vaccination. Can we vaccinate and take care of this? So they make a peptide against a conserved epitope on the phage. It's conserved across many different isolates, and they immunize mice with this peptide. They show that the mice make antibodies, and then they infect them. They make a wound and put PA in it. And they see that vaccination reduces the incidence of wound infections by about half, which, it's an interesting effect. It's not what you would want from a vaccine, but presumably you could improve on it.
Well, I slapped my head when that statement came out. I said, this is remarkable that a vaccine against a bacteriophage protects you from a bacterial infection.
Mm-hmm.
Vaccinating against a virus of a bacterium protects the human or the mouse host from a bacterial wound infection. This is incredible.
Certainly, it's counterintuitive, right?
Well, they didn't optimize their peptide construct for the fact that it's only halved is quite remarkable.
I'm sure they can
absolutely.
improve it. I'm sure
that's
they can
a great day in the lab if you see a 50% drop.
You don't have to have a statistician to say, is this real?
Yep.
I mean, this looks, think about this, because Pseudomonas is a really nasty infection for not only wound victims, but burn victims, as well as cystic fibrosis patients. And if you could come up with a vaccine against a bacteriophage, and obviously they have to do the next experiments and ask the question, are biofilm-based infections in the lung with Pseudomonas aeruginosa colonized with filamentous bacteriophage, or more importantly, whether vaccination of a mouse would actually confer protection against a lung infection? That would be headline news for the cystic fibrosis.
So
community.
You would say that they need to look in the lung infections to see if the phage is playing a role, right?
Yeah.
Certainly, because cystic fibrosis, there's a large burden of disease, but burned patients are frequently get contaminated with Pseudomonas. So that's a huge application, and it seems to me that would be a lot more accessible wound to try this therapy with. Yeah,
it's an easier animal model.
Yeah, well, you'll be happy to know that this lab is actively pursuing the vaccine and use of monoclonal antibodies, which, Vincent, you didn't mention, but they also showed that antibodies against the phage would protect. So they're pursuing clinical application of this work.
Yeah, the antibody experiment's interesting. They make monoclonals, they give them to the mice. They are protective against infection, but they don't, these antibodies, which are against the phage, they don't block phage internalization, but they seem to promote phagocytosis of lysogenic bacteria. And they say maybe there is phage on the surface of these bacteria, and the antibody's coating it, and it's opsonizing the bacteria, right? And
well, filamentous phage bleb off.
Yeah.
They bleb off.
Exactly.
And so it's not unusual.
Yeah.
I believe the phage receptor is also a pilus. I believe they make that point. So you could imagine a bacterium with appendages that themselves are decorated with phage, which then become a target for the antibody. So now the whole bacterium gets ingested. But when I talked with Johanna, she said this was an incredibly exciting day when they added the antibody to their in vitro experimental model, and were just holding their breath to see what would happen. And they were just so happy.
They had to go out and buy champagne.
So, Michael, they say in the discussion, our findings may complicate efforts to develop phage as a therapeutic option.
Oh, yeah. Oh, yeah.
But they say, however, to date, lytic phage have not been reported to suppress phagocytosis. So maybe there's a difference between lytic and lysogenic phage, right?
And it is the lytic phage that have been applied in therapy strategies.
Right.
So I think it's too early to say that these lysogenic phage will also, that it may not be relevant.
Well, one of the problems with lytic phage is that they're so rapidly cleared by the reticular endothelial system. They're rapidly cleared. And so, like I said, I spent 2 years.
That's a different problem about what we're talking about here. Here, the problem is the phage themselves.
No, I do appreciate that.
The immune response. It's a different, look, there are plenty of problems with phage therapy, right?
Yes.
We don't need to enumerate them right now. What is the case is that this is news.
Yes.
That the lysogenic phage does what they say it does. That is news.
And boy, I think they knew that this was going to be quite a startling and fascinating biology. I think you all will agree with me. They did not leave many stones unturned. This was a really comprehensive
There's lots
study.
of figures. There's lots of figures.
Well, and really rigorous immunology, careful cell biology.
Yeah, it's very well done for sure.
Yeah.
And there's methods. You can see how they did stuff. They put methods in the paper. My goodness.
Did you want to wind this up, Vincent, or shall I go ahead with the profile of our lead author?
Go ahead, Michele.
So Johanna Schwer is this was her thesis research in the lab. She hails from the Netherlands and always was interested in the natural world and curious about how humans work, how do people get sick. So she pursued as an undergraduate at Utrecht College in the Netherlands as kind of a standard pre-medical, biomedical curriculum. But she realized that it was the mechanisms that really fascinated her rather than treating the disease. So she decided to pursue a master's in microbiology and immunology. She got some experience with Enterococcus, but also had an opportunity to do a kind of an externship. She went to Boston and worked with an immunologist there at Harvard Medical School, and that really exposed her to immunology. And she decided then to pursue a PhD in immunology and to stay in the U.S. So that is what brought her to this Stanford lab. She said that this was a really ambitious project, and she credits her advisor for recognizing when they had a question that they needed the answer to, but that it could be done much more efficiently and expertly if they reached out and recruited a collaborator that had expertise. So that's why we see so many different authors. So, for example, she wanted to really credit Patrick Secor, who was a postdoc at the time, who really taught them the phage biology. He's now a faculty member at the University of Montana. And then they also collaborated with immunologists. They had people doing taking samples from burn wounds from patients to start the project. She had the benefit of working with a talented undergraduate, Michele Bach, who helped her with the animal experiments. And these are laborious, tricky surgeries, a lot of animals. So it was really a group effort. And what we get then is this really impressive, collaborative, and rigorous paper. I did ask what advice she might have for other people who are pursuing a career in science. And she was reflective and said, at the beginning of your training, it's just impossible for you to fathom how much you're going to learn and how much you're going to grow during that process. And you need to be open to that, realize that you're going to be a different person when you come out with your advanced degree. And what she really appreciates is, and she learned this from advisor, is when you hit a technical barrier, rather than, bang your head against the wall or feel like you've got to learn the technique, like reach out, find experts, get collaborators, have other mentors, and do this team science, because it makes the science better, it's more efficient, and you end up with this group that can become your colleagues, for the rest of your career. Now, while she was doing this amazing work, Johanna was also curious about how can I get science really helping people? And she took advantage of really some, I think, groundbreaking programs that are available at Stanford for their trainees to take classes in a variety of business classes. So, for example, she took one in negotiation and influence, innovation and management in healthcare, drug discovery and development project that was done in collaboration with Novartis, another one on preparation for biotech business and finance. Wow. And while she was doing this really great collaborative research, so she defended in January of 2019, and she is now doing consulting with a firm that focuses on biotech and life sciences. They help these companies do the research and analyze the data so that they can better understand how to get their medicine to the people who will benefit the most. So she's had a really interesting career, and we had a great conversation. Congratulations again, Johanna, on this beautiful work.
Lovely. Very nice. Thank you, Michele.
Yeah. Oh, and she also, by the way, when she's not working, enjoys hiking and being outdoors and spending time with friends out enjoying the beautiful outdoors there in Northern California.
Nice. All right. Let me read a couple of emails. Elio, this one is for you. This is from Melissa, who writes, I recall Elio mentioning in a past podcast there were human lab members who had drank E. coli K12. Do you happen to have a reference for this? My colleagues and I are really interested to read about how this idea came about.
No, I'm sorry, I don't. I can look into it. It's old stuff.
It's old stuff.
I remember that story too. I think it became part of microbiology lore.
Yeah.
It was probably from the 20s/1930s and it was probably done on the West Coast where K12 came out of a lab at Stanford, I think. And I remember hearing the story that, people drank it and nothing happened. And it was bandied about at the time of the recombinant DNA controversy of making Andromeda strain like microbes, and people were very much concerned about it. And the Asilomar conferences resulted from the fear, and it was principally fear of the unknown, as this was brand new technology that people didn't quite understand.
All right. Next,
sorry about that.
No problem. Next one is from Josep. Hello, everyone from TWiM. Hope you are all having a pleasant day. I'm a graduate of medicine and health sciences from a small university in Marília, countryside of São Paulo State, Brazil. I'm very passionate about science and especially micro and molecular biology. So as a newcomer of the show, I must say you made my moments extremely happy. On the last episode, moreover, I got very excited. That was the one which you did, Michele, about extracting nutrients from E. coli, I believe.
Through type 3 secretion. But he's talking about the snippet, I believe.
Is he? The snippet was bacteria store energy for gutless worms.
Yes. Yeah. Elio's one.
Fan mail aside, I wanted to compliment Michele when she asked how the Napoleon worm-bug symbiosis started, because I had asked myself this question when you started the snippet. Maybe there is a specific location for the worm to procreate, and the habitat where its early life develops is associated with bug contact. Also, when Vincent joked about who's in charge, I remembered this magnificent excerpt from Yuval Harari's *Sapiens* on, quote, was wheat or us in charge of the agricultural revolution, which begged the thought in my mind, it is well known the importance of the enteric nervous system on physiology and behavior of the human. Now how much of it is controlled by gut bugs?
There's always bias involved. We'll just say the gut bugs control all of us.
Anyways, thank you very much for your work. Please continue. Best regards. Thanks. All right. Thank you, Josep. Karen writes, Dear TWiM team, thank you for presenting the absolutely fascinating story of the flatworm Paracatenula and its chemosynthetic symbiont. Mind expanding. There was one point that needs clearing up. At 2109, the question was asked as to whether the endosymbionts are vertically transmitted. The paper gives the answer. Quote, each species of Paracatenula harbors a specific CA regiera, and the endosymbionts have been vertically transmitted for at least 500 million years. Such prolonged strict vertical transmission leads to streamlining of symbiont genomes, and the retained physiological capacities reveal the functions the symbionts provide to their hosts. My comment, 500 million years, that is way back in the history of animal life. A paper
a beautiful response.
A paper that
very much. It's lovely.
A paper that describes the reproduction of Paracatenula provides an understanding of how this vertical transmission occurs. Quote, most catenulids predominantly reproduce by a type of asexual fission termed paratomy, during which new clonal worms grow from the posterior end of the animal, thus generating a chain of zooids. The Latin word catenula means small chain. The zooids subsequently develop into separate individuals and detach from the mother zooid. Sexual reproduction is rarely observed in catenulids and has never been observed in paracatenula. The paper provides evidence that when the zooids form, they contain bacteriocyte cells that house the endosymbionts. Thank you. Thank you.
Oh, this is lovely. Thank you so much.
Very, very thoughtful.
And one more. Mark writes, you seem to be an enthusiastic supporter of vaccination, and I agree with you, but how to best influence reluctant parents remains an issue. I agree. There was a letter in the Wall Street Journal a few days ago that I thought you might find interesting. The complicated truth about vaccines. Dr. Michael Siegel writes that vaccines, can appear to be the proximate cause of a condition when they have nothing to do with the ultimate cause. The letter focuses on a 2010 study published in The Lancet that aimed to establish whether the apparent association of Dravet syndrome with vaccination was caused by recall bias, and if not, whether vaccination affected the onset or outcome of the disorder, and finds in Dr. Siegel's words that vaccines did trigger the deterioration, but had the children not been vaccinated, the same deterioration would have occurred anyway the next time they had a cold. Dr. Siegel summarizes the study by studying the detailed time course in a genetically characterized population. They illustrated how vaccines can appear to be the proximate cause of an injury, yet the ultimate cause is genetic, and concludes that unless we act like scientists and explain the science, we won't be effective in helping laymen put the frequently reported vaccination horror anecdotes in context. This is a very good point, because a lot of parents say, my child did so-and-so after getting a vaccine. And we all know the vaccine didn't cause it, but we can't give a plausible alternative, right? So if you can't, the parents don't believe you. And so this is a good point where they were able to provide a plausible alternative.
Population studies kind of address that.
For sure. For sure. I mean, when you immunize 100 million people, things are going to happen, right? Because things happen anyway.
And we don't yet know all the precise triggers of how the immune system reacts. And remember, everyone's immune system is unique to them.
Yeah.
And it's I often tell the students that the immune system is the original cumulative exam. You're, exposed to everything from the time you are born.
But that's not the target. We're not talking about students. We're talking about lay parents.
I
That's
understand.
a very different problem.
I understand that, and I agree with Mark. We have to come up with a way of explaining the vaccination horrors that they are viewing in their children, or they're hearing from the neighbor down the street. But we have to explain to them that the harm of not vaccinating their children is far worse.
Well, that's the problem.
It is a problem. I had an argument on Twitter a couple of weeks ago. Someone said, after being vaccinated, my son never spoke again. And, I know the vaccine didn't do that, but I can't tell her what did it. And unless you can, they're not going to be convinced. It's tough. It's very tough. All right, that is TWiM 198. You can find it on any good podcast player. If you listen on a phone or tablet, please subscribe to the podcast so you automatically get every episode. And we know how many people are listening. And as I said earlier, if you enjoy what we do, consider supporting our work. You can go to microbe.tv/contribute. And we have a couple of different ways you can help us out, including giving a small amount of money every month, $1. And less than a cup of coffee, you get not only TWiM, but TWiV, TWiVO, TWIP, and Immune. 5 science podcasts, probably more than you can listen to in a month. But wow, for $1, what a deal. Microbe.tv/contribute. And of course, send your questions and comments to us at TWiM@microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at the wonderful blog Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent.
And Michael and Michele will join me next month at ASM Microbe in San Francisco, where we will be doing a TWiM. So check us out.
For a live audience.
Live audience. I'm Vincent Racaniello, and you can find me at virology.ws. I want to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.